we're asking really hard questions about how can we freeze and transport biological material, so cells, tissues, proteins and bacteria. The way we got into this was there's a really cool protein in fish which live under the Arctic Ocean, and they make proteins which have sugars on them and they stop ice crystals from growing. And as proteins are just biology's polymers, I thought that we could combine our expertise in sugar chemistry and polymer chemistry to try and mimic how these fish stay alive. So we want to be able to make a difference in regenerative medicine, where you can grow and replace parts which have failed. And the big challenge if you try and achieve this is if you have, say, a kidney from one person, you have to get it to the other person intact and still working.
A lot of antifreezes used at the moment are chemicals, and they are very, very, very poisonous, whereas these proteins have evolved in animals, so are generally quite safe. These proteins are incredibly difficult to work with. They are hydrophobic, which means you can't get crystal structures for them with current technology. They're also unstable above certain temperatures. They evolved in fish in Arctic waters, and they don't like being warm. You do run into the problem when you're trying to modify the gene that it's incredibly stable and it doesn't take modifications very well.
There's a lot of therapies people are looking at where they take cells from a patient, they grow them up in a lab and they can inject them back into the patient, and it might be for immunotherapy and some cancer treatments. The problem we have is that you might not want the cells when you take them out of the patient. You might want to store them. At the moment, cell storage is quite difficult. So say you froze 1,000 cells, you might only get 500 back. And obviously for the patient, they need all 1,000.
So while we look at ice and how you can store cells and tissue, the key things we use to do this is synthetic polymers. So one of the polymers we use most in this is essentially a derivative of wood glue.
So we're making the additives that people are likely to use when they store these organs. So I generate a lot of polymers and ampholytes that when mixed with blood or organs will help to prevent the ice crystal damage.
So last year we did a project co-sponsored by NASA. And what we did in that was using a high altitude weather balloon that goes to the edge of the Earth's atmosphere, we sent some biological samples up, including some blood. So the blood alone, when it went up to the outside of space, it froze, pop, 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 the cells didn't survive. We added in some of our new materials, these protected the cells from the freezing, so when we recovered the balloon, when it crashed back to land, we could see that the cells were intact.
We're still at sort of the initial stages right now where we're just proving that we can modify this protein. Once we've proved that we can make these modifications, it will then be running through a list of them to see which modifications are best.
We've now created a set of design rules that might help us rationally design some of these materials, and I think taking that forward and making the best materials we possibly can. If the NHS didn't have to get rid of organs, then the transplant list would be a lot shorter, and that would enable us to save a lot of people.